My name is Dominic Sabani, and in this final project, I will be predicting gene expression in response to environmental stress using variational autoencoders. Climate change brings increased risks to crop resilience. Farmers around the world will have to adjust to new extreme weather events. However, given these weather events are increasingly sporadic and novel, it would be useful to know how the genes of crops will respond a priori. We focus on rice. Rice is a staple crop for around half the world's diet, and we aim to answer the following question. Can we predict how the gene expression of rice will change under different environmental stressors? We have access to over 3,000 rice accessions, which we align to the rice genome and then quantify using feature counts. We also have metadata associated with each sample. This metadata contains information such as the treatment, experimental details, and also the place of origin. Since these accessions were obtained in a variety of different conditions from labs across the world, it is unorganized and not immediately amenable to being encoded. We use a language model to categorize the experimental conditions for each sample and are left with conditions such as cold, drought, and flood that we can then use downstream to train our model. Our model is built on top of a variational autoencoder, which takes a sample input X, and in our case, gene expression data, and outputs a prediction of that same sample. It has two main components. First, there is the encoder, which takes the input and encodes it into a latent space, and then the decoder, which samples from that latent space and tries to predict the same input X. We built a variant of a variational autoencoder to compare the baseline VAA to, called the conditional variational autoencoder, which has the added feature of concatenating the environmental metadata to the input, allowing the model to learn how genes might vary across different environmental conditions. The variational autoencoder is optimized using the evidence lower bound, which is enabled by the reparameterization trick, which allows the gradients to flow through the model. Elbow prioritizes maximizing the likelihood of the data while also ensuring the approximate posterior does not diverge too far from the prior. In our evaluation, we focus on a couple metrics, mean squared error, Pearson, and Spearman correlation. Pearson correlation measures the linear relationship between the actual and predicted gene expression for a sequel sample. The Spearman correlation evaluates whether the relative ordering of gene expression levels is preserved between the actual and predicted sample. We find the conditional variational autoencoder performs better than the vanilla VAE across a variety of different experimental and environmental conditions. While the VAE achieves slightly better mean squared error for a couple conditions, the conditional variational autoencoder performs better on Pearson and Spearman correlations. By conditioning on the environmental metadata, the conditional variational autoencoder can better distinguish gene expression patterns that are specific to individual environmental conditions. This results in a more accurate and context-sensitive latent representation, enabling the decoder to generate precise gene expression profiles. There are multiple different next steps from this project. Future work could involve collaborating with researchers in the field and seeing how the model's predictions relate to real world experiences of how these crops will actually deal with environmental conditions. Additionally, incorporating quantitative environmental treatment levels, such as the magnitude of the intervention. So we could include varying intensities of temperature or drought and flooding and evaluate how the gene would respond to these conditions. Thank you.